Okay, so today we'll talk about this gene called ACAT1 and two genes that we've seen several times before, TREM2 and LRP1. The paper that we're discussing today is titled, and here's a mouthful, Inhibition of Acyl Coenzyme A Cholesterol Acetyltransferase 1, that's the full name of ACAT1, promotes shedding of soluble triggering receptor myeloid cells 2, that's TREM2, and low-density lipoprotein receptor 1, LRP1, dependent phagocytosis of amyloid beta protein microglia. All of that will make a little more sense towards the end. I noticed, I hadn't noticed at first, but the senior author in this paper is the senior author in the RIN3 BIN1 paper that we discussed a few weeks ago at Harvard that we emailed and they replied very, very gently and so forth. So that's at least the PI seems to be the same in both papers. Okay, so in the introduction to the paper, the authors bring our attention to the fact that the discoverer of Alzheimer's disease, Dr. Alzheimer, originally reported the accumulation of lipid droplets in glia of AD patients. Dr. Alzheimer basically sliced the brains of his patients who he noticed were developing this very particular kind of dementia and looking at them under very rudimentary microscopy. And yes, he did notice the accumulation of the A beta plaques and the tau NFTs, but he also noticed the lipid droplets in glia. Right? So that led to a whole bunch of people interested in what might be the connection between lipid metabolism and Alzheimer's disease. And previous work showed that this enzyme, ACAT1, that catalyzes the esterification of cholesterol and produce cholesterol esters, which are more easily picked up by cells and out of circulation. What, in relationship to ACAT1, what previous research showed is that patients who suffer late onset Alzheimer's disease have increased cholesterol esters, and their microglia have higher levels of ACAT mRNA. The microglia in these patients are producing more ACAT1 mRNA, which presumably would lead to the formation of CEs, which would then be picked up by neurons in load patients, right? So far it's all about what ACAT1 does to these cholesterol esters and how they're producing in higher or lower levels. But in parallel, previous work in vivo has shown, and from their lab, has shown that inhibiting ACAT1 could reduce the A beta plaque formation and could increase A beta clearance by microglia. But the mechanisms through which this happens remains unclear. Right? So we have on the one hand this connection between ACAT1 and cholesterol, and some epidemiological connection between CE levels and ACAT1 levels and Alzheimer's. On the other hand, we have some previous work saying that ACAT1 manipulations can change A beta plaque formation and A beta clearance. And lastly, they bring to our attention that TREM2 is this receptor that is expressed primarily in microglia, promotes cholesterol metabolism and AB clearance, right? So what the authors are doing is sort of putting two and two together and saying perhaps ACAT1 somehow works through TREM2. And that's the hypothesis that they decided to explore in this paper. Okay? This is one of these papers in which there's some previous evidence for this, there is some previous evidence for that. And we know that this molecule might be connecting the two. Let's take a look. And that's what they're doing. So first, they wanted to confirm that ACAT1 could actually be inhibited by a drug called AV. So ACAT1 is an enzyme. It's a acetyltransferase. So it has enzymatic activity, and you can inhibit it pharmacologically. And there's this drug AV that can inhibit ACAT1. Now, the question that I have for all of you is, if you have a drug that you think would inhibit an enzyme, how could they confirm that AV works in microglia? It had been tested probably in other cell systems, but they wanted to test it in microglia. So how would you do that? What do you look for? CE levels. That's right, because the known catalytic enzymatic activity of ACAT1 is transferring acyl groups to cholesterol. So you just look for what this thing does biochemically normally, and you look at CE levels. Excellent. So that's exactly what they did. And they took microglial cells in culture. It's this BV2 line, and for now, these are wild type BV2. We'll see that that changes later. And they did an AV treatment, and this is a dose curve in a way, or a titration experiment in which they have CNT, is how they denote controls throughout the paper, and they add increasing doses of AV to the cultures, and they measure on the y-axis cholesterol ester levels, and they see that with increasing doses of AV, the amounts of cholesterol ester drop, and they drop significantly. In a way, we got here by you guys answering a question of how scientists could confirm that their drug functions as expected. Why else would scientists conduct these sorts of pilot experiments that focus on unrelated readouts? So this paper is not about CEs. So why would the scientists want to know what happens to CEs in these cells other than confirming that AV works in these cells? Hershey?
So they get cited?
So that they get cited as okay. Interesting. So the argument here is that they're doing an experiment that others may want to communicate via reference, saying, well, Tanzi et al. And their group show that AV can lower the levels of CEs in microglia, and then they take it from there. Interesting. I hadn't thought of that, but I'll take it. Fair enough, but that's what we, in a way, already said, right? That this is just to confirm that the drug is working to lower the levels of CEs, right? So confirmation, it's reassuring, but that's sort of the first reason that we discussed. The mechanism of action, so how do you derive the mechanism of action from this? Fine, but this experiment, would this experiment show you any of that? This experiment stops at saying you inhibit the enzyme, you get less of the product that the enzyme normally makes. No receptors, no anything. That's the key. So what happens so often is it's not so much, I sort of introduced this experiment as how could you confirm that AV works as expected in microglia, right? And what you do to do that is check for like some sort of low-hanging fruit readout, like formation of the product that this enzyme normally makes. Before you start asking questions about biological effects, phenotypes, and so forth, you just want to make sure that you're inhibiting the enzyme. And to make sure of that, you simply look at the product normally produced by the enzyme. But one thing that these experiments also offer is some sort of sense for thresholds of sensitivity, because this AV drug may be going easier into certain cell types than others. It may be absorbed more easily or less easily by certain cells than others, may be metabolized by some cells more than others, and so forth. So what the authors gain from this is also a sense for sensitivity. And what do you think? So what dose would you not use based on these results? One, right? One micromolar. Their own results is already telling them that they can't even see a drop in the enzyme's product. So right off the bat, this tells them, well, you have to go higher than 1. And it also shows them that there is some sort of what appears to be some sort of dose dependency that is pretty linear, meaning that the more drug you add, the more inhibition of the enzyme you see, which is somewhat expected. Okay? So that's right. So we're confirming the effectiveness of the drug in this particular cell type, and we're getting a dose curve out of it so that we can identify thresholds. So they went for phenotypes, and remember that what they're interested in is the potential function of this enzyme ACAT1 in internalizing A-beta peptides and clearing them. So A-beta peptides are typically cleared by microglia in the brain, so they're They're interested in how much microglia will uptake and clear A beta peptides, right? And how that depends on ACAT1 function. So they subject the same cells to the same treatment, but this time they look for internalization of A beta 42 peptides, the really toxic A beta peptides, added to the media, right? And they see that it's only at 10 and 15 micromolar that there is a significant increase in uptake of the peptide. So my question to you now is, why do you think these responses or these response profiles are so different from one another? For inhibition of the enzyme at the catalytic level, we seem to have that linear regression going down. For inhibition of or enhancement of AB peptide uptake, I suppose, there is this lag followed by a very steep increase in the function. So why do you think that might be? So you're saying that around 10 there is a different pathway that is triggered? Oh, I see. So what you're saying is that this is not a response to ACAT inhibition itself, but it's a response to the levels of CEs. That when it reaches that threshold value, that's what triggers. Okay, interesting. Any other ideas? Beautiful. So now we're combining both. So what we're saying, or what you guys in a way are saying is, Lucy's proposal might explain why effects don't kick in until you reach the 10 micromolar dose of AV when it comes to A beta internalization. That the cells may need to go below a 75% rate of CE for them to say, okay, now we need to take up AB. But then Gabby's point is, okay, that explains the threshold aspect of it, but how do we explain the fact that the levels of CEs are decreasing linearly with those, but then after this threshold, the AB internalization goes up geometrically. In fact, it seems like it maxes out. So it goes from nothing to everything. It goes from none to all. And you're absolutely right. What normally explains that are mechanisms that kick in or introduce these positive feed-forward loops or positive negative loops, actually, as well, that will say, okay, we were holding off past this threshold. We need to turn this thing on, and we need to turn it to the max, right? And that happens a lot when you compare the effect of drugs on the catalytic activity of an enzyme, because that's a very one-to-one, it's a very straightforward reaction formula, if you will. You have the enzyme, you have the substrate, and now by adding the inhibitor, this pharmacological inhibitor, you're keeping the enzyme from turning substrate into product, right? So it's a very linear one-to-one relationship. There are no loops, positive feedback loops, feed-forward loops, or logical gates, or anything of that sort. Whereas when you're looking at internalized A beta peptide, between the activity of the enzyme that is just sticking acyl groups into cholesterol molecules and internalization of the A beta peptide, which we know is a very complex multifactorial process by which this endocytosis, sorting of the endosomes, and so forth, between those two things, there may be so many things that include a whole bunch of loops and mechanisms that will make the response become all or none. Okay? This seems silly, but this is one of the keys behind that ages-old discussion between how do you merge the fact that molecules in cells and in organisms behave analogically in that they're floating around, bumping into each other. And then when you take a step back and you look at the way that cells and organisms and tissues behave, there seems to be a certain computerized logic, or Boolean logic, we'd call it, right? Where things are ones and zeros, they happen or not. And then there's these regulatory gates that seem like a computer program, right? Condition A and condition B, then boom. Condition A or condition C, then boom, right? And everything is so all or none, right? How many times we talked, for instance, about cells that either are alive or they decide that they will die, but rarely do you see cells that are half dead or something like that, right? That there are some, or when in processes of differentiation, we've talked about microglia, astrocytes, and oligodendrocytes, and they all come from the same precursor. But we've never really spoken, except for one exception that I remember, we've never really spoken of cells that are somewhere in between, not knowing what they are, right? Because biological processes are full packed with these loops that will make them either not respond to something if we haven't really reached a threshold, or go all the way if we have. And this is a beautiful example of that. So we have that threshold that appears to be around 75%, call it ester, or that correlates with it. We don't know if it is the 75% of CEs that trigger this, but it correlates with going over that threshold of ACAT inhibition. But once we go past that, boom, the cells will go, quote unquote, all in. Okay? Questions or comments, anyone? So now we have that ACAT1 inhibits A-beta uptake in microglia. How do we know that? Because when you inhibit ACAT1 with AV, A-beta uptake goes up, as long as you give them 10 or 15 micromolars or more. So that's what they see, that's what we see. And what the authors think is that that may be mediated by TREM2. So they know that TREM2 is important for uptaking A-beta. So what they're hypothesizing is that ACAT1 is inhibiting A-beta uptake through inhibiting TREM2. So how would you test the effect of AV? Or how would you test if it is true that the effect of AV is through TREM2? So we add AV, cells that have TREM2, and cells that don't have TREM2. And then what we expect is AV has an effect here, no effect here. What have we called these before, sort of colloquially? Right? Taking out that middle person, right? Taking out that intermediate. That's not the only approach. What other approaches can you think of? Okay, so when you say you start without it, you mean cells that don't have TREM2, and so maybe these cells here, and then you reintroduce it. But then what are you looking at? So in a way, you're proposing the same experiment, but looking backwards. So it is the same experiment. But there is a cousin of the experiment that you proposed. And that is, you guys remember? Sometimes when people hypothesize about an intermediate that is mediating the thing, they say ACAT1 is, let's say, inhibiting TREM2, and TREM2 is important for a beta uptake. So what's another prediction that you could make? Overexpression of what? You gotta make a choice. TREM2, there you go. Yeah, so anytime that we have these hypotheses about a sort of like a two-step process where cause A is working through intermediate B and causing effect C, taking out the intermediate is usually a really good experiment, and sometimes just saying, well, if it is through this intermediate that things are happening, if I add a lot of the intermediate, or I wake up that intermediate, or I somehow activate the intermediate, I should see the same thing. And that can be done through an overexpression of TREM2, in this case, the intermediate. So that's valid too. What's the problem with that experiment though? What might be a problem of saying I'll take cells, microglial cells, I'll put more TREM2 in them, and I want to see if they uptake A beta better or not. You're not showing the relationship between AV and TREM2, fair enough, yeah. You're simply saying TREM2 can do this thing, doesn't mean that it's the intermediate of AV, I like that. What's the other problem? The other problem is, in the context of this paper, that's all news, right? It's in the introduction already. We all know that TREM2 facilitates A beta uptake by microglia. That's how they got here. So for them to say, we'd like to see if overexpressing TREM2 will increase the uptake of A beta, it's almost like saying, dude, you told me that it did, right? So they actually, even though formally speaking, it is typically an approach that people take when they don't know any better. In this particular case, this second half had been established already, that TREM2 can induce A beta uptake. What was more important now is, as Melita was pointing out, and in a way Arshia was reinforcing, showing that it is through TREM2 that AV functions to induce A beta uptake. Okay? I think I got everybody. Yeah, so that's exactly what they did. They took wild type cells. In this case, they worked with human cells. These IMGL cells are induced microglial cells. So these are cells that were induced to become microglia from some sort of pluripotent stem cell line. And they had two versions of them, cells that are wild type or cells in which the TREM2 gene has been knocked out. And what they see is that when they add AV, that's the red bars, to the wild types, there is this significant increase in A beta uptake, as expected and as shown before with the other cell line. But when they treat the same with the same AV knockout, TREM2 knockout cells, there is no change. So we took out the intermediary and we killed the ability of AV to induce A beta 42 uptake. So what that is saying, what that is supporting is this idea that yes, TREM2 is the intermediary, you take it out and now AV can't do anything to A beta uptake. For now, they'll go with ACAT1 somehow inhibits TREM2, right? And that's why when we take out ACAT1, TREM2's activity would be somehow increased, and that's what explains the increased A beta uptake. The question now is, what is ACAT1 inhibition by ways of AV doing to TREM2 in these cells? So in the paper, they write this. I paraphrase, but they write this. We first thought that AV treatments would lead to higher TREM2 levels. That treating the cells with AV would simply increase the levels of the TREM2 protein. But what they found is that the TREM protein levels, shown in this first graph here, with AV decreased significantly. When they looked by Western blotting at how much TREM2 these cells had when they were treated with AV, they had less. But in a parallel experiment, they also saw, using ELISA to look at things that are secreted into the extracellular medium of these cells, they saw that when these cells were treated with AV, there was a significant increase in extracellular TREM2, something that we've called soluble TREM2 or S-TREM2 before. So the conclusion from these two experiments is that ACAT1 inhibition decreases TREM2 in cells, but increases the levels of soluble TREM2 floating around. So from that, the authors, for now, conclude that ACAT1 inhibition might promote S-TREM2 shedding. Now, when the authors are presenting these results, if you read through the text, it's interesting how they say, well, you might think that that's a bad thing. We just said that from the introduction, we've been telling you that TREM2 facilitates A beta uptake. The more TREM2 cells have, the more A beta they can uptake. Now we're telling you that the levels of TREM2 in the cells is decreasing, there is more floating around. One might think that's a bad thing. It turns out that previous work has shown that soluble TREM2 can bind to A beta peptides that are around cells and bring them to the cells and facilitate the uptake, right? So with that prior observation in hand, which they hadn't made, they cite someone else, they say maybe for these cells to have more S-TREM2 floating around is a good thing, right? So they decided to go with a working hypothesis that AV is inducing an increase in the levels of S-TREM2 in CM. Notice how I changed ACAT1 to AV now. So treating the cells with AV is inducing the shedding of S-TREM2 in the conditioned media of these cells, and that is facilitating the uptake of A beta. STRAM2 was shown to prevent A beta peptides from oligomerizing and may serve as a chaperone. So that's the working hypothesis that soluble TREM2 can take A beta peptides in the extracellular medium and accompany them to where they should be so that they can get endocytosed. And to test this idea, they decided to conduct this experiment. They plate the cells on day 0. Let's do red. They plate cells on day 0. Then they treat the cells for a day or so with DMSO, which is the vehicle for drugs, or the drug itself, AV. Right? So they're treating the cells for about a day with AV or no. They let the cells react to that AV and put out to their culture media whatever they're putting out. Now, if the cells were treated with AV, what do you think this medium will be full of or have higher levels of? What will be increased in this culture media of cells treated with AV? That's normally added from the outside. These people think that it's there on the slide. So what will go up in the culture media or the conditioned media of cells treated with AV? Exactly. So in the control cells, well, these are going to be control conditioned media, right? But for the AV treated cells, this is going to be conditioned media with a lot of TREM2. It's important to understand that to understand the rest of the experiment, because what they say is that then they will collect those media, which can be quote-unquote spiked by STRAM2 in the AV treated cells or not in the controls, and then treat another batch of cells with those conditioned media. So what they're doing there is, can the conditioned media of one group of treated cells affect the function of a different group of cells? Is there anything in this conditioned media that can affect the ability of other cells to then, here also is the A beta, they add the A beta and then they do the usual, they count how much is internalized, right? So what this experiment was, in a way, testing is this idea, if the hypothesis is true that AB leads to a lot of soluble TREM2 in the conditioned media, and if it is true that that is what's causing the increased A beta uptake, then by simply taking conditioned media from AV treated cells and using it to bathe naive cells, we should see those cells being better at uptaking A beta now. Because what they need, STRAM2, it's there already. So that's what they did. They forget AV now. They're treating cells with only conditioned media from either control cells or AV treated cells. And what they see is that when these are control cells in both cases, right? Both are called CNTs, control cells. When they're treated with control conditioned medium, there is a baseline level of A beta uptake. But when they treat those cells with conditioned media from AV treated cells, now A beta uptake goes up significantly. Once again, I can't emphasize this enough. These cells have not been treated with AV. They've been treated with the conditioned media from cells that were treated with MV, but they themselves were not treated with AV. So this goes with, in a way, Sebas, if I may go back to your point, this is in a way testing the idea, so you go straight through the intermediate. If this three-step process or I guess two-step process with three things involves AV through STRAM2 leading to AV A beta uptake is true, then if you shortcut the thing, if you short circuit the pathway and you simply add STRAM2, then you should see that A beta goes up, right? A beta uptake goes up. But that's what they're showing in this experiment. But then the question becomes, is that all AV does? Put stuff in the CM? Or could AV be doing something else to cells? In other words, for now we're going with you add AV to microglial cells and they start pumping out a lot of STRAM2, which might facilitate A beta uptake. But is that all AV does? Or could it be acting through parallel pathways as well that simultaneously contribute to increasing A beta uptake, right? Or could AV do something else? How can you check? How would you check if AV could be doing something else? AV, making AV fluorescent? The A beta? That's how it's done already. We didn't discuss those results, but the internalization of A beta 42 is done through the usual mechanism of you make A beta peptides fluorescent and then you follow the fluorescence. The question here is, how would you check if AV, in addition to producing a lot of S-TREM2 in the media, might be doing something else that further contributes to A beta internalization? Beautiful. So what you're saying is you add AV, you expect that S-TREM2 induction. That's what the model proposes, and that will lead to A beta uptake. But what you do is you come here and somehow take it out. Okay. All right. What else could you do? Any other ideas? You could, but that's taking the question to the next step, right? So you're asking about mechanisms. Here we're at a very initial phase in which we're simply asking, is AV doing everything it does through S-TREM, or could it be acting through additional parallel pathways? So in other words, could it be acting through some other mechanism that goes around and also contributes to A beta uptake? I like that. So what Ali is saying is let's add soluble TREM2, right? And if the induction of A beta is just as much as you get with the conditioned media, it means that S-TREM was the only thing that was doing this thing. But if it's less, the difference should be accounted for by something else that AV is doing. Okay, I like that. Any other ideas? When it comes to these questions of, is my original treatment, in this case AV treatment, doing everything through one intermediate or through more, right? The approach this group took is similar to approaches that we've seen before. And the idea here is as follows. If it's true that AV is acting through S-TREM2 and something else, then a way to dissociate between these two is to say, okay, if I'm adding S-TREM2 alone, right, I'm going to get some effect. But if I add AV on top of that, I'm going to further increase the effect because now I'm going through the parallel pathway, right? We go back to this idea that whenever you think that there is only a linear path of causes and effects, then adding earlier steps won't really change things much. Whereas on the other hand, if you start from some sort of middle point and you go back and you add the earlier step and you see an increase in the outcome, that's probably an indication that there is something else that is going on the sides, right? And that's exactly what they did. What they said is, okay, let's take these cells that are treated with conditioned media from AV cells, so theoretically are bathed in S-TREM2 and whatever else AV is making cells produce and secrete. But in addition, let's treat them with AV to see if they respond further. Because if these cells responded further, it meant that AV is doing something to the cells that has nothing to do with things that the cells produce and secrete out. But that's not what they saw. They saw that adding AV to those cells did not further increase their ability to uptake A beta. So the conclusion from that for the authors was AV treatment induces A beta 42 uptake fully via soluble factors released into the CM. And notice the changing language. I'm not saying through soluble TREM2 anymore, because what they moved from one dish to the other was not soluble TREM2. It was the whole conditioned medium. So it could be soluble TREM2, it could be something else, it could be a combination of both, right? So how do we know if it's only TREM2 soluble TREM2. How can we check? And this idea was kind of already thrown out. Jacob. Oh, okay. So how would that work? I think I know where you're going, but what would that look like? So you're saying STREM2, but not the, okay, cool. So I think I know where you're going. So how would you do that operationally? I like the idea. What Jacob is saying, let me paraphrase, is if we suspect that AV is inducing the secretion of other things other than STREM2, and those may contribute to A beta uptake, how can we have a medium that was not treated or doesn't come from cells treated with AV and yet has a lot of STREM2 in it, right? And an operational way of doing that, one is to take culture media and spike it with STREM2. And as we'll see in a second, that's what they did. But what you're saying is a little finer, a little more elegant. And your idea might be to take conditioned media from control cells and add STREM2 to those. In other words, saying, okay, I'm going to take conditioned media from cells that were never treated with AV, so they were secreting and pumping out whatever cells normally pump out, and to that, I'm going to add a little STREM2. If AV was producing and pumping out something else, then I shouldn't see an effect with this control condition medium plus STREM2. If STREM2 was the only thing that AV was pumping out that mattered, then I should see that I can phenocopy the AV treatment. So your proposal is slightly more elegant, but what they did is a little simpler. They simply took 40 nanomolar concentration of recombinant soluble TREM2 that they added to the media, and they incubated the cells with that, with a STREM2 spiked culture media, and they saw that the A beta uptake was significantly upregulated. So that's one way. That's what I call their option one. What might be another way? If you suspect that, well, but maybe AV is forcing the cells to pump out a bunch of things. STREM2 is one of them, but there are others. And I actually don't think that STREM2 matters that much. What could you do to try and test that idea? Excellent. Take out that STREM2 and see if the AV conditioned medium still works or not, right? So what they did is this. It's a little more nuanced. But what they did is this. They took wild-type cells, right? They treated with nothing, and there is 100% internalization of A beta 42. Then they treated them with AV, and these are normal cells, so obviously there was more A beta internalization. They've been showing us that all along. Then what they did is they treated with AI, not what you think. AI here is an ADAM 1017. I didn't think that you would remember, but ADAM 1017 inhibitor, right? So what they're doing there is basically inhibiting the ability of cells to produce STREM2. In a way, it's also your idea, right? Let's incubate the cells with AV, let them do what they normally do except shed TREM2, right? And when they did that, when they added the ADAM 1017 inhibitor, there was no increase in A beta uptake. That's fine, but what really matters here is that when they added AV plus the inhibitor, now those cells did not show an increase in A beta uptake. Now those cells could not uptake A beta better. Even though they're exposed to AV, if you don't let those cells shed TREM2, they won't uptake A beta. And then the last one, which is interesting, I forgot to, I had shown this bar before. It's all part of the same graph. But the last group, which is interesting, is everything together. AV, the ADAM inhibitor, and soluble TREM2. And why is that last control important? Why is that? So when they added everything together, yes, now the cells are uptaking A beta just fine now. Why do you think that last control is important? What is that control telling us? Yes, that sure, the inhibitor kill the ability of cells to make STRM2, but if you add it from the outside, if you add it exogenously, as we would normally say, it still works. So the cell is still responding to STRM2. What we did with the inhibitor is kill the ability of cells to make STRM2 and therefore kill the ability of AV to do anything. Okay? But if you add STRM2 on the side, the cells are responding just fine. Very bluntly put, that's in a way of saying these cells are alive, they're still functioning, they can still internalize A beta, you flooded them with drugs, inhibitors of ACAT and ADAMs and whatnot. So these cells are being bombarded, but yet if you add some soluble TREM2, they will pick up A beta. It's just the machinery is working, it's just that it wasn't being turned on. Okay? Beautiful. And option 3, in the interest of time, I think, because you already mentioned it already. So we go back to this idea of add AV, right? And you suppose that AV will be pumping out a whole bunch of things, among them STRM2. But you also suspect, or you want to make sure that STRM2 is indeed the one responsible for this and not any of the other things. How else could you take STRM2 out of the way? They did it by adding the ADAM inhibitor. How else could you take STRM2 out of the way? We've seen this approach before. Whenever you have something floating around, how can you block something that is floating around? With blocking antibodies, right? And I can't remember, but we may have seen papers in which they use blocking antibodies against soluble TREM2. Soluble RP1, LRP1, I'm pretty positive, but TREM2 as well. So yeah, they could have treated the cells with AV, and they could have gone back to that beautiful conditioned medium experiment. So they could have treated the cells with AV, let them release everything that they normally release into the conditioned medium. Then you take that conditioned medium and you fill it up with STRM2 blocking antibodies. Now you don't let STRM2 do what it normally does, and then you treat cells with that, and what you should see is that the cells are not internalizing A beta anymore. Cool. So we have a few to go. I think we'll make it. Just like the authors knew about TREM2 being important for A beta clearance, the absolute sort of star of A beta clearance in the brain is actually LRP1. Right? LRP1 is the one that clears A beta from the brain by pumping it into blood and whatnot. So they started entertaining the possibility that AV may actually work through LRP1 as well to induce A beta uptake. So to start testing this, first they said, okay, let's look at that first leg of the pathway. Can AV treatment induce LRP1 activity somehow? And lo and behold, when they treat cells with AV, there is a significant increase in LRP1 protein levels. Cells are making more LRP1. So that kind of tests the first leg. The second leg is, okay, let's treat wild-type cells with AV, as we normally do, right? Significant increase in A beta uptake. But now, let's take cells that don't have LRP1 and see if they respond to AV just the same, and they don't. A beta uptake stays slow. So that means that for the AV-induced A beta uptake, LRP1 is required. Seems to be one of the intermediaries, right? That leaves us now with two competing models. It could be that AV induces LRP1. LRP1 somehow induces the shedding of TREM2, and that is what is causing A beta uptake. Or it could be that AV somehow induces the shedding of TREM2, and that is what's acting to induce LRP1, and that is what in turn causes A beta uptake, right? What experiment could distinguish between these two, Arshia? So you're saying adding LRP1 or STRM2? Okay. So how do you start at LRP1? What do you mean by we start at LRP1? I think I know where you're going. Yeah. So you're all in a way saying the same thing, right? So let's start with one. Let's activate one or induce one or overexpress one. Let's take out the other, and vice versa, and let's see which one conforms to the predictions, right? And that's exactly what they did. What they did is they said, okay, we're going to treat the cells with soluble TREM2, right? So that's what we're adding to the media. They already had that. That's easy. We'll add soluble TREM2, but we'll add it to cells without LRP1. So they added it to LRP1 knockout cells. And remember all those previous times that soluble TREM2 increased the uptake of A beta? When they offer soluble TREM2 to cells without LRP1, no difference. In fact, what appears to be a mild decrease in the uptake. Okay? So now cells without LRP1 cannot respond to STREM2. So what they're favoring, they didn't show the counterpart. They didn't add a lot of LRP1 and take out soluble TREM2, by inhibiting ADAM or whatnot. But with this evidence, they think that it goes like this. Inhibiting ACAT1 will inhibit the shedding of STREM2, which in our updated network model, I'm saying ACAT1 normally inhibits shedding of TREM2. And that shedding of TREM2 somehow facilitates the uptake of A beta through LRP1. And because LRP1 is uptaking A beta peptides from the extracellular medium, that will lead to inhibition of A beta oligomerization. The reason I have this here as oligomerization instead of the peptides is because that's the node that we already have in the network, right? And then I plugged in ADAM 1017 in there because it was already in our network, and it is contributing to the formation of STREM2. But in a separate paper many weeks ago, we saw that ADAM 1017 could also inhibit LRP1.